Welcome to Living Well with MS. This show comes to you from Overcoming MS, the world's leading multiple sclerosis healthy lifestyle charity, which helps people live a full and healthy life through the Overcoming MS program. We interview a range of experts and people with multiple sclerosis. Please remember, all opinions expressed are their own. Living Well with MS is listener supported. If you'd like to support us and help keep our show advertiser-free, donate online at overcomingms.org/donate. And now, let's meet our guest.
Stefano Pluchino. Stefano received his MD and PhD degrees at the University of Siena in Italy and has had additional training at Cambridge University in the UK. He's currently Professor of Regenerative Neuroimmunology and Honorary Consultant in Neurology within the Department of Clinical Neurosciences at Cambridge University. He's here today to talk about his research in advanced cell therapy. So, welcome, Stefano.
Thanks so much, Jeff, for an invitation.
I hope you're enjoying weather in the UK. It's
very windy nowadays.
It's somewhat, yeah. I'm interesting that you've moved from somewhere that I'd consider almost paradise to where we live. But anyway. So to start off with, could you introduce yourself and your work and your interest in MS?
Yeah, actually, you already pretty much introduced me, but I can elaborate a little bit on my background. I am clinically trained as a neurologist, but I've always had a very, very strong interest in experimental research and preclinical research. And I have spent, I would say, the great majority of my career trying to identify first and then to develop new therapies for MS. I started with my PhD in experimental neuroscience, where we develop an experimental therapy made of brain stem cells for an animal model of MS in mice. And then we moved to a larger non-human primate study. And as you can see from the most recent publication, which I think is one of the reasons why we are today, we finally managed to complete a first-in-man phase 1 proof of concept human study using the very same principles, but of course in humans using human brain stem cells for people with progressive MS. So that's my background, that's where I'm coming from, and that's my interest.
So the trial was specifically for progressive MS. So firstly, how would you define progressive MS? And how is it different from relapsing-remitting MS or common form of MS?
Yeah, there is multiple ways to define progressive MS. One of which is, which I think is very well understood even to lay people, is a type of disease which is characterized by accumulation of neurological dysfunctions in the absence of clinically evident flares of disease. The definition, the neurological definition is a little bit more complex than that, because we are using clinical and imaging readouts to identify what is progressing and what is not progressing. What we know, the more we understand about MS, is that within the same nomenclature, there is a very heterogeneous type of disease, which might differ from patient to patient. But in general, just to be clear, whenever we identify progression in the absence or in presence of clinical relapses, then we start discussing of progressive MS. There is a distinction between a disease which is characterized by the accumulation of indolent progression from the very, very beginning, from the onset, which is what we call primary progressive MS. And there is also a disease which instead is an evolution of an initially relapsing-remitting type of disease, which is called secondary progressive MS. And then, in the context of primary and secondary progressive MS, we can distinguish, and it is becoming more complex, between active and non-active types of primary or secondary progressive MS, where activity is attributed to either clinical episodes of disease, or radiologically evident episodes of disease, which can be identified by the use of contrast agents at the time of the MRI. So very, very complex, very heterogeneous type of disease, with or without activity, which inevitably undergo accumulation of neurological deficits.
And most treatments are for relapse-remitting MS, aren't they? So
yeah, in the general belief or in the general knowledge, we are very lucky. We and patients are very lucky to be having the opportunity to be considered or offered for what we call disease-modifying therapy. So basically, the pharmacology of MS has to distinguish between two main categories of drug treatments. The former of which is for symptoms, what we call symptomatic treatments, with minimal impact on the disease progression, on the disease evolution. And then we have what we call DMTs. DMTs is an acronym which stands for disease-modifying therapies. So these are therapies, I would say, next-generation biologics or advanced therapeutics, from which we expect a modification of the disease evolution, or what we call trajectory. And in the context of these DMTs that we have available on the NHS, but which are under continuous development and discovery in clinical trials, the great majority of what we have available at the moment is for relapsing-remitting MS, because the initial exercise to develop disease-modifying therapies for MS was targeting that type of inflammation which leads to clinical relapses, new hospitalization, increased costs on the NHS. But what's becoming very interesting is that since a few years, there is a general worldwide, I would say, consensus that progressive MS is becoming and has become a clinical unmet need, and the whole MS world is concentrating money, finances, funding, and efforts towards stopping MS. And stopping MS through the development of a new generation of DMTs, which will eventually target progression rather than relapses.
And do you think ultimately that could work for all forms of MS? Or will there always be a sort of relapse-remitting DMTs and progressive DMTs?
Whenever you have to identify a potential drug pharmacological target and develop a new therapeutic, you are inevitably bound to a good understanding of what we call disease pathobiology. So what is the mechanism of disease that you want to target? And what is becoming more and more accepted is that mechanism of disease in progressive MS, both primary and secondary, are very similar to disease mechanism in other classical neurodegenerative conditions, including, for example, frontotemporal dementia, Alzheimer, Parkinson, and other chronic conditions of the brain. So there is expectation that this exercise of identifying new targets or repositioning FDA or fully approved medication from another neurodegenerative conditions to MS might have a good impact both in primary and secondary progressive MS because of the shared mechanism of disease.
And so your trial, could you tell us a bit about the trial design and how you gathered the participants and selected the participants who ultimately took part?
Yeah, the trial that you are alluding to is the one that we published back in December last year, which received lots of attention in the media and in the scientific community, and which is the beginning of the end, actually. It is the end of 25 years of work preclinically, and it is the beginning of another journey, which will be that of showing non-ambiguously that there is space for advanced cell therapies for progressive MS. So it did take as much as 20 to 23 years from the initial first experiment in mice to complete a phase 1, first-in-man, dose-escalating proof of concept human study in people with progressive MS. The clinical trial design is very simple because it is similar to many other phase 1 studies. And to understand the clinical trial design, we have to remind ourselves what is the remit of a phase 1 study and the ambition of a phase 1 study. A phase 1 study is the first human trial in the long journey leading to authorization and licensing of a new medication for clinical use. And the aims of the clinical trial are usually the feasibility of the study, the safety of the procedure and the administration of the drug, and the tolerability. So everything but means of efficacy. There is no placebo control group. There is no control group whatsoever. But what is important is that we assess very rigorously whether we can produce a new medication for clinical application, whether that medication is not putting at risk the health and the life of participants, and the medication is well tolerated. And I'm very, very proud and very happy to say that the trial that we completed reached and met all these major aims. It was doable, it was safe, and it was very well tolerated.
But of course, I was going to say the other thing, which you might have been coming on to then. Did you also test for what results you got? Whether so. I mean, that's the first thing. Obviously, it needs to be safe. But then, sort of leading up to further trials, did it also have an effect on the participants?
Yeah, that's what conventionally we do in phase 1 studies. So we try to develop a little bit of biology or biomarkers in the trial, without emphasizing too much on the outcome, because there is no control group, but which is a piece of evidence that we use to start putting together a phase 2 study, which is the inevitable follow-up of a successful phase 1 study, aiming in this case at measures of efficacy. And we did indeed a little bit of much more than a little bit of biology in the phase 1 study by collecting longitudinal biofluids from participants. So actually we did two things. The first thing that we did was to escalate the number of injected cells from a minimum of 5 million cells per patient to a maximum of 24 million, in order to identify what we call maximum tolerated dose of cells. And there were no adverse events whatsoever, regardless of the number of cells injected. So which means that even 24 cells, we can escalate even further. And then we collected, as I was saying, biological fluids multiple times across over 12 months of follow-up from patients. And the way we managed to do this sampling, I think, is second to no other study, because thanks to the incredible support of those participants to the trial, we were able to collect 7 longitudinal serum samples in 12 months and 5 longitudinal CSF samples. So these individuals, the participants to the study, underwent 5 consecutive lumbar puncture from the beginning to the end. And then we subjected the CSF and the serum to what we call untargeted, unbiased, high-throughput profiling, looking for metabolites, for proteins, and for lipids, in order to study the way the metabolism of the brain responded to the transplantation of the advanced cell therapy product. And that's a biology that we started gathering, because regardless of the intrinsic limitation of the study, one of which is the fact that we were, for ethical reasons, we were bound to recruit people with very advanced disease who were not eligible for any other treatment. So it is a very late stage disease with very high levels of disability, with hypothetical minimal response to a given treatment. Regardless of this, we found very interesting biological readouts in the CSF and plasma, which are very, very important, very promising when putting together a phase 2 study. And the type of response that we collected was that still, despite the very late stage disease type and the very advanced disease, the brain was responding to the treatment in a dose-dependent and time-sustained fashion, which means that the more cells injected, the more response, which was sustained over 12 months of study follow-up, which is very, very exciting biologically and moving forward.
And you've mentioned the difference between primary and secondary progressive. This was just for secondary, is that right? But is there likely to be an expansion of the trials for people with primary progressive MS as well?
In full honesty, there was another I don't want to call it competing. It was a complementary independent trial which was conducted in Italy, in Milano at San Raffaele Scientific Institute, which enrolled people with primary and secondary progressive MS, active and non-active. So it was much more heterogeneous in terms of patient selection. We decided to be more homogeneous, and we recruited and selected people with active and non-active secondary progressive MS only. But I think moving forward, there is enough room to expand some of these preliminary early findings in human also towards primary progressive MS.
And so you said stage 1 was successful. So is a stage 2 trial planned?
Indeed.
And how will you select participants for that trial? If people who are listening are interested, how could they apply to be part of it?
Of course, there is no website yet available, or there is no MS registry yet in place where a patient can apply. We are working very, very intensively towards an international consortium, which would include representatives and key opinion leaders from main European and non-European countries, including the UK, to establish a phase 2 NSC transplantation consortium. And by the time we will reach a stage of maturation of the project, including some funding, we will make a press release and we'll make people aware of the direction the consortium is taking and the different possible means by which they can apply to be considered for the study, to be screened for the study, and then to be eventually enrolled into the study. Inevitably, a phase 2 study has to have some prerequisites which make it very different from a phase 1. So one of these requisites would be to slightly review the type of patients to consider. So the disease has to be a little bit less advanced. And there would be a placebo control group, because otherwise it would be equally very difficult to extrapolate any means of efficacy. We are planning to expand towards an higher dose of cells, so 24 million, starting from 24 millions and considering also twice as much cells, because there were no adverse events at 24 millions of cells. And there would be lots of imaging applied to the patient matrix, because by means of sophisticated imaging, we might infer whether the applied ACT, advanced cell therapy, is impacting on the disease trajectory by which mechanism, which can be neuroprotection, reduction of inflammation, remyelination eventually. But we need imaging, because otherwise it will be very difficult.
So there's a potential, because you just mentioned remyelination there, which I think is a sort of, I mean, people who've had MS for a while, that's their kind of dream, because you think, well, okay, you can stop progression, but there's that dream of actually getting improvement. Is that a potential then, if you found signs that it might be remyelination?
I think the potential is there. I'm a great believer of multiple mode of actions, because one single mode of action, I think, is not enough to interfere meaningfully or in a way which is clinically relevant with the complexity of MS. MS is not a simple disease. It's made of multiple mechanisms of disease which are redundant in time and space. There is regional diversity, and there is also temporal evolution of MS. There is space for assessing whether a proportion of the injected cells are able to differentiate in vivo into new myelin-forming cells. There is a space to assess whether some of the cells are able to increase the survival of neurons in the brain, and there is space to assess whether the transplanted cells are able to reduce the type of inflammation which characterize progressive MS, which is what we call now smoldering inflammation. This highly diffuse, homogeneous, low-level activation of microglia and astrocytes, which might be reduced by means of ACT. And the reason why I am specifically alluding to these three major biological mechanism of disease, remyelination, reduction of inflammation, and neuroprotection, is because we have managed to identify each of these mechanisms in clinically relevant animal disease models in the last 25 years. So there is expectation that a clinical trial designed in a way that it will be allowing us establishing the efficacy of the treatment will reveal what we have established already in animal disease models.
And so when we say stem cell treatments in MS, I think most people would probably mostly think about HSCT or hematopoietic stem cell treatment, or possibly autologous stem cell transplant. So those are sort of traditional, if you like, but the stem cell treatments which are familiar, and they use chemotherapy to effectively wipe out your immune system and rebuild it from stem cells. So is this a similar sort of action, or is it distinct from those systems?
I think it's very distinct, and the distinction is based on the different biology. What you call HSCT, or hematopoietic stem cell transplantation, or bone marrow transplantation, is a procedure which was established for blood malignancies back in mid-60s. So it is already kind of 50, 60 years old. Established technology which has the ambition to reset the immune system of the individual receiving the transplantation. Without going into very much technical details of how autologous HST is done in people with MS, we know now that unless there is disease activity, disease progression is not much affected by the transplantation of autologous hematopoietic stem cells. What we have developed is an advanced cell therapy, which is basically made of brain-specific stem cells. So these cells are extracted out of miscarriage, human fetal material. They are fully characterized in the laboratory, and then they are expanded, increase in number, put on proliferation and duplication in the presence of very complex recipes of chemically defined media, and then they are prepared for transplantation. So the idea is to put back in the brain stem cells of the brain for neuroprotection, reduction of inflammation, and remyelination. The HST go to the bone marrow. So the site where the transplantation of bone marrow stem cells engraft is the bone marrow. The site where the stem cells of the brain should be engrafting and surviving is the brain. So there are two completely different approaches.
So you're not using chemotherapy?
We are using a very, very low dose regime of immune suppression for precaution.
Okay.
Because the way the phase 1 study was done was to use a single miscarriage human fetus as a starting material, and immunologically that fetus is recognized by the immune system of the receiving patient as allogenic. So we need to avoid the risk of rejecting the transplanted cell. So it's completely different from the intense chemotherapy that is given to people in preparation for the bone marrow stem cell transplantation. This is a very low dose combination of immune suppressant to reduce the risk of rejecting the graft.
And you mentioned it's a miscarried fetal donor. Is it potentially possible that you could actually get the stem cells from the patient themselves in the future?
Yeah, there is two options actually. One is the one that you just mentioned, but we have, and other groups have data that would suggest that there is something wrong in the stem cells of the patient. So at least there is something wrong in the body of the patient, which might predispose to the risk of developing MS. So we have to be very careful when proposing autologous therapies. The other option, which is very, very doable because of the fantastic technology we have available, is to use what we call cell reprogramming methodologies to generate brain-specific stem cells from minimally invasive biopsies from the skin or from the blood, from the hair follicles, out of which we can make, using, again, cell reprogramming technologies, unlimited numbers of tissue-specific stem cells, including brain stem cells. What we call iPSC technology. This is a Nobel Prize in Physiology and Medicine 2012 to Shinya Yamanaka and John Gurdon, which is now applied in the clinic to make pluripotent-like stem cells and to derive the tissue-specific stem cells from these pluripotent stem cells. So it is doable, and there is technology available.
That sounds like science fiction to me. So you're literally taking them from any cells effectively, and you can build specific stem cells from that.
Right.
So if, assuming everything was to go well in phase 2, then there's normally a phase 3 trial before actually normal patients, if you like, see the therapy. So how long would that typically take to go from where we are now at the end of phase 1 up to actually a DMT going on the market?
This is what everyone.
How long is a piece of string?
Yeah, this is what everyone would like to know, including myself. I think it would take less than 25 years. I think it will take less than how long it has taken to bring the first experiment to the conclusion of phase 1. Usually we can do clinical trials in 2 or 3 years and make very elegant prediction of outcomes in the long run. But the key thing is that phase 2 and phase 3 clinical trials, especially for advanced cell therapies or advanced therapies in general, are extremely expensive. So the academic work that we have done so far, because this is a fully academic journey, would be unlikely to be fully sustainable if there is no strategic partnership with either a funding body or a society or pharma, because that's what becomes game changing in terms of speed of delivering the outcomes when moving to larger trials. So that's, I think, the key limiting factor. 2 things are the source of the cells, so we need to refine protocols and methodologies to make it fully sustainable and fully renewable, and then the funding, because once you get to large human studies with lots of patients, with hospitalization, MRIs, biomarkers, and the cost become insane.
And so what you're sort of saying, 10 to 15 years would be a typical.
I think it's going to be less. If the phase 2 trial is successful, then I think the interest will raise exponentially, and with the interest might be also a raise of the available funding or the number of people interested to partner with the consortium.
Mm-hmm. And just to try and understand a little bit more about how it works, could you explain how you go about reprogramming microglia, and how changing the brain's metabolism and fatty acids could halt disease progression?
Yeah, that's another thing that we are doing in the lab, which does not apply directly to the ACT trial. That's a very, very elegant and innovative piece of mechanistic science that we are doing in my lab in Cambridge. We've been studying actually how cells behave in physiology and in disease, and together with other groups, we have discovered that the initial, if you like, old-fashioned understanding of cell types, there is B cells, T cells in the blood, there is microglia, astrocytes. So we believe there is a single cell type. It's completely wrong. And each cell type has multiple, what we call cellular states. So basically, a single cell is able to transit between multiple different functional states, from homeostasis to disease. And all this is regulated by their metabolism, the way they use the food that we provide, or the food that the organ provides, or the blood provides. And by studying very, very molecular aspects of how metabolism works, we have identified a specific metabolic feature which is associated with a specific type of microglia that is responsible for disease progression in MS. And by targeting and by knocking that function down, we are able to stop progression in animal models. And we are able to save neurons. So basically, we have identified a potentially new pharmacological target, which is specific for microglia, which is accessible via brain-penetrating small molecules. So it is pharmacologically druggable, and which plays a major role in the damage to neurons, which is responsible for disease progression.
And for this trial, are you at the start of the 25 years?
This is not a trial. This is
It's all the research,
mouse experiment
Mm-hmm.
with very, very sophisticated genetic tools and small molecules and transgenic mice, very, very sophisticated and single cell analysis, which is the beginning of another journey that we hope will be faster than the stem cell one, leading hopefully to another trial with a molecule which will block microglia from becoming bad and from becoming responsible for disease progression in MS.
So, I mean, I think the takeaway from that would be that that's going to take quite a long time. But there's constant research, though, isn't there? It's like it's not like, okay, there's one new drug. It's like there's actually loads of trials happening, and here we're just talking about. I mean, it's a leading university certainly, but a university in a country, and presumably you mentioned Italy as well, but presumably around the world there are many centres that are doing other research. So.
Correct.
It's a hopeful.
Correct. And there is no Stefano Pluchino only in the world. There is other groups who are contributing to advancement of knowledge and develop a new understanding of mechanism of disease and identification of new drugs. So everyone is working to make it possible and to allow everyone saying that we can stop MS soon.
And just as a final question then, what would you hope that your legacy is and the impact of your research?
But the legacy that I envisage is students coming to my lab and developing their career as independent scientists and traveling in the world and creating this network of individuals with enthusiasm and ambition and curiosity to identify why the MS brain goes wrong and how to fix it. And this is something which is happening already. There is people who have been trained in my lab and scientists who have been trained in my lab who have been traveling all over the world. They are back in other universities. They have been offered academic faculty positions and they have established their own groups. And this is already ongoing.
Okay, so I think just to finish, just to wrap up, I'd say if people are interested in as someone who's been classified now as secondary progressive MS, I think is absolutely fascinating because you do feel a little bit left out in that there's a huge choice of treatments for relapsing-remitting MS and very few for the progressive types. So if people are interested, the thing to do would be really just to at the moment, keep an eye on information coming out about future trials. Is it likely to happen in the next year or so that you would start to be talking about phase 2 trials?
There is lots of trials already ongoing. There is trials mostly driven by pharmas. Mm-hmm. One key example of a phase 3 trial, which is currently ongoing in the world for people with progressive MS, is the BTK inhibitors trials, which are being run by multiple big pharmas at the same time. There is also academic trials. I think we have in the UK the largest ever academic trial for progressive MS. The name of the trial is Octopus. Mm-hmm. It has received funding, or at least it's been negotiating funding with the UK MS Society, multi-million £. It is currently recruiting. So I think the impression of being left all out is understandable, but I would say it is temporary because of the number of efforts that the whole community is putting around progressive MS. We should remember, everyone listening today, that there is an initiative which was started a few years back, which is called Progressive MS Alliance. This is a worldwide alliance uniting MS societies and scientists and companies to attack progression in MS. So we will get there. And I think it will take much less time than how much it did take to attack relapsing-remitting MS. Don't forget that what we see now in the NHS, all the DMDs, were started being developed back in mid-90s, and they were not available until beginning of 2000. So it did take a while already at that time. Now, relapses and inflammation is curable in MS. Progression is not curable yet, but everyone is committed to understand why progression happens and how to stop it. And it will happen.
And so if people are interested in trials, in my personal findings, that actually my MS nurse put me onto a research group. I live in the southwest of the UK, and it was a southwest research group, and they then gave me the contact details for the Octopus trial. So I would say, and I think it's probably worldwide, if you speak to your healthcare professionals, they would typically have the connections to be able to set you up with trials if you're interested.
Yeah, I think the pathway is always the same. Your healthcare professional, lead neurologist, MS nurse, and then MS society. Because you'll find any information you're looking for very much up to date in the MS Society website, or MS Society chapter, or MS Society branch, or MS Society working group. So I would say lead neurologist, MS nurse, MS Society.
Okay. And with that, I'd like to thank you, firstly, very much for your research, and secondly, for joining us on the podcast.
Thanks, Geoff. It was very pleasant to speak with you today.
Thank you.
Thank you for listening to this episode of Living Well with MS. Please check out this episode's show notes at overcomingms.org/podcast. You'll find useful links and bonus information there. Don't forget to subscribe to the podcast so you never miss an episode, and please rate and review the show to help others find us. This show is made possible by the Overcoming MS community. Our theme music is by Claire and Nab Dean. Our host is Jeff Alex. Our videos are edited by Lorna Greenwood, and I'm the producer, Regina Beach. Have questions or ideas to share? Email us at podcast@overcomingms.org. We'd love to hear from you. The Living Well with MS podcast is for private, non-commercial use and exists to educate and inspire our community of listeners. We do not offer medical advice. For medical advice, please contact your doctor or other licensed healthcare professional.